Today's video is going to be all about cell membranes, which is Unit 2, Chapter 7 of Chemical Biology in AP Biology. So let's get to it. Hi everyone, my name is Mikey from Avo Prep Academy, and today we're moving on to cell membranes, which is a really important chapter in setting things up for the next unit where we talk about photosynthesis and cell respiration. So we want to have a really great understanding of this chapter. This chapter 7 is primarily divided into two parts. The first part is going to be about structures of membranes and different proteins that are associated with it, and then the second part is going to be about transport of materials across that membrane. So we want to focus on both of those components today, with a little bit more emphasis on the second part, of course, because that's the primary thing that we need to know in moving forward. However, let's begin with part 1, which is about membrane structures in general. So plasma membranes or cell membranes are very important because they're found in every single cell, and every organism has at least one cell, meaning that this is a feature found in all living things. Now, plasma membranes are primarily comprised of what we call phospholipids, which are special amphipathic molecules, which means that these molecules are at once polar and nonpolar. And what happens when you add a bunch of phospholipids into an aqueous solution is that you get the formation of what we call a phospholipid bilayer. Now, the phospholipid bilayer has tails that face each other and heads that are facing outside towards water and inside towards water. And this separation is incredibly important in the things that we're going to talk about today. So now that we have the primary component of the cell membrane, we need to understand that there are other substances that are associated with the cell membrane other than phospholipids. And this is what we call the fluid mosaic model. The fluid mosaic model stipulates that yes, there is fluidity about these phospholipids and the proteins that are embedded within them, but the mosaic part is where we discuss the proteins and all of the other associated molecules that are found within the plasma membrane. So let's begin with the fluidity part first. All cells have to retain some homeostatic level of fluidity. Imagine a red blood cell traveling through a very tiny capillary. It needs to be flexible to fit through the small diameters of these tiny vessels that we call capillaries. And as such, these cells are going to be flexible. However, in some cases, that fluidity can become compromised. For example, in cold temperatures where everything becomes rigid, cell membranes can become rigid. At warm temperatures where everything becomes more fluid, cells can also become more fluid. Now, cells have two ways of battling these environmental conditions. One is that cells can control the level of saturated fatty acids found within these phospholipids in order to increase or decrease fluidity. Let's take a look at this example. Here we see on the left side a cell membrane with a bunch of phospholipids with unsaturated fatty acid chains. If you remember, unsaturated fatty acid chains have kinks, which then gives these phospholipids a little bit more space in between them, allowing them to become more fluid. So in cold temperatures where everything starts to become rigid, then the cells are going to want to increase the amount of unsaturated fatty acids within these phospholipid bilayers. On the other hand, the right image shows a bunch of saturated fatty acids as found in these phospholipids. This reduces the amount of empty space between the phospholipids, allowing the fluidity to become slightly more rigid. Now, this would be useful in warm conditions where the cell becomes too fluid, and the reduction in spacing in between them can give them some rigidity. Now, another way that cells can control fluidity is by the addition of cholesterol molecules. Now, this is only found in animal cells, but cholesterols do something really cool. At cooler temperatures where things get rigid, the presence of cholesterol molecules give spacing in between the phospholipids, allowing them to remain a little bit more fluid. On the other hand, when temperatures get warm, the fact that the cholesterol molecules are holding onto lipid tails on either side give them that rigidity. So it sort of acts on both ends to retain that level of fluidity somewhere in the middle. Now let's focus on the mosaic part of the fluid mosaic model. Mosaic means that we have a heterogeneous system, as in that the cell membrane is not simply a monolith of phospholipids, but rather have materials that are embedded within them. And one of the main things that we need to know about are these proteins. In this image, we see integral proteins, we see peripheral proteins. We also see the cholesterol that we've just mentioned, and things like glycolipids and glycoproteins. The ones that we want to focus on in this chapter are these integral proteins. Integral proteins are associated with the lipid bilayer and it integrates into the membrane. But the ones that go completely across the membrane get a special name, and those are called transmembrane proteins. And when we talk about membrane transport, proteins that can move things in and out of the cell are going to be by default transmembrane proteins, and those are going to be the ones that we're going to be focusing on. And while the chapter does introduce a bunch of membrane-associated proteins with various functions, the only function that really matters for the context of this chapter is this transport function. So now we get to move on to part 2 of chapter 7, which is about membrane transport. We can't really start membrane transport without talking about a very important phenomenon in our universe that we call diffusion, though. So let's get to that first. Diffusion is this idea that materials will move from an area of higher concentration to an area of lower concentration. And of course, we're talking about that specific solute's concentration. And in this image, you'll see that we have a bunch of orange molecules on the left side of this system. And there is a membrane here, but we can sort of ignore that for the time being because it clearly shows that these materials can move past that membrane. And what you'll see over time is that these orange molecules will move from where there are more of it to where there are less of it. And they do this until they reach what we call a dynamic equilibrium. It's called dynamic equilibrium because it's not that these molecules are not moving at all. They are constantly moving, but the concentration is going to be relatively consistent across the entirety of the system. But what's really fascinating about diffusion is that each solute can diffuse in accordance to its own concentration. As in, if we also add a purple molecule to the right side of this tank, then each of the orange and the purple molecules is going to move until each of them have reached its equilibrium. So it's almost like they know what they are and they're going to act in accordance to their own concentration. And the directionality of this movement is usually referred to as moving down the concentration gradient. You can think about concentration gradients like a hill. If you have a ball up at the top of a hill, it will simply move downwards without any energy. And likewise, these concentration gradients allow materials to move from where there is more of it to where there is less of it without the input of any energy. However, you could imagine that if you were to try to get that ball from bottom of the hill to the top of the hill, that you would have to invest some energy. And that's exactly what happens here too. If you want to go against the flow of the universe, as in go up against the concentration gradient, then you'd have to invest energy as well. And we're going to talk about that towards the end of this lecture. Now that we have diffusion out of the way, let's first talk about passive transport. As the name suggests, passive transport is the movement of materials in and out of the cell without the need for any energy. And because we've talked about diffusion just now, you should be familiar with the idea that when we talk about passive transport, all materials will be moving from an area of high concentration to an area of low concentration along or down the concentration gradient. And here we're going to talk about two main types of passive transport. The first is called simple diffusion. Simple diffusion refers to materials that are small and nonpolar, completely ignoring the cell membrane as it moves across it. So when we have materials like oxygen gas or carbon dioxide gas, these are very small and nonpolar substances. And these materials will simply move from an area of high to area of low concentrations, completely ignoring the membrane altogether. And as such, we just refer to these as simple diffusion. However, there are materials that require some help in moving across the membrane, and that is due to a very special trait of cell membranes called selective permeability. So let's talk a little bit about selective permeability. The name suggests that the cells can decide which materials can move in and out. And unlike those small nonpolar gases, most materials that are dissolved on either sides of the cell membrane are not going to be able to pass through the cell membrane itself. And the reason is that, as we mentioned earlier, the outside environment is aqueous. The cytoplasm is aqueous. Materials that are dissolved in aqueous environments are typically polar and charged substances. And because polar and charged substances cannot pass through a lipid tail bilayer, what happens is that the materials that are on the outside stay outside, and the things that are on the inside stay inside. However, this might not be the best thing for the cell, especially if you want materials to be exchanged. So in order to accomplish this, cells perform something called facilitated diffusion. Facilitate means to help, and in this case, the help is coming from those transmembrane proteins that we saw earlier in this video. You see, transmembrane proteins that are involved in passive transport come in two varieties: channel proteins and carrier proteins. Let's talk about the channel proteins first. Channel proteins are literally tunnels that are built into the membranes via these proteins. And these tunnels can be open or closed in accordance to the cell's desires. But the important thing is when the channels are open, it's going to allow very specific molecules that are allowed to pass through that channel to pass freely in accordance to their concentration gradient. And an example of this would be something like a sodium channel. And in this image, you'll see that a sodium channel being open would allow sodium ions to move from the outside to the inside with no issues nor energy associated with it. Typically, sodium ions should not be able to pass because it is charged, but through these membrane-bound proteins, they can. The second type of proteins that are associated with facilitated diffusion is called carrier proteins. And in the case of carrier proteins, these are proteins that have the capacity to uptake a material from an area of higher concentration and then move it to an area of lower concentration by the way of the movement that is found within the protein itself. It's almost like Pac-Man, which then opens his mouth and closes his mouth, allowing materials to move from one side to the other. Now, in comparison to channel proteins, you can imagine that the rate at which materials can move could be a little bit slower, but these are both going to be useful for membrane transport. I do, however, want to interject and make a very important point here. You can see that as these materials are moving from an area of high to low concentration, this Pac-Man-like carrier protein is moving. And we know that any movement at all is going to require energy. But we said that passive transport doesn't require any energy. So where is this energy coming from? The idea is that when you have a higher concentration gradient of materials on one side, as they come to the other side, the area with lower concentration, this movement itself can release kinetic energy. And that is what we call the chemiosmotic gradient. And the chemiosmotic energy that we get when things are moving from an area of higher to area of lower concentration can provide sufficient energy to move these carrier proteins. Now, this concept is going to be important for later on. Now that we have passive transport out of the way, we're going to take a sidestep here and talk about a very specific type of passive transport that we need to know for this chapter, and that is called osmosis. Now, osmosis is diffusion of water. However, the problem with the idea of osmosis is that typically we think about movement of materials in accordance to their concentration gradient, but you can't calculate a concentration of water because the water itself is a solvent that we input into the overall calculation of concentration to begin with. And it's also important to note here that water has a free pass through the cell membrane. And this is not because water is nonpolar. Of course it is polar, but it's because we have these very special proteins called aquaporins that are present in almost all cells in varying amounts. And aquaporins allow for free passage of water through that plasma membrane. So in this case, what we have to be concerned with is the direction in which water moves. Because if too much water comes into the cell, it could potentially swell up and burst. And if too much water leaves the cell, it could shrivel up and die. So what really determines the movement of water? Well, the answer is what we call osmolarity. Now, osmolarity is slightly different than what we call concentration of solutes. And here is how we can demonstrate this idea. Imagine a U-tube, not like the type that you're watching this video on, but an apparatus that's shaped like a U. If we fill this U-tube with water and put a membrane in between the two sides that are permeable only to water but nothing else, well, let's take a look at what happens as we add solutes to one side of this U-tube. See, the thing is, usually the solutes are going to want to move to the other side, but because of this impermeable membrane to the solutes, it cannot do so. So if the solutes can't move to change the concentration, what can? The solvent. And in this case, water will move into the area where there is a greater solute concentration in order to attempt to equalize the concentration on either side. But the important part to note here is that the solute concentration is not based on a single solute, but the totality of all of the solutes within a system. And that is what we call osmolarity. Now, in the case of cells, because they have aquaporins, it mirrors this U-tube example perfectly. Because cells are going to have a certain amount of solids dissolved within it. It has DNA, protein, sugars, and all of the other organelles. Now, the external environment, whether it be oceans or freshwater ponds, are also going to have solutes that are dissolved within it. And the direction in which the water is going to move is going to be dependent on which of the two sides have greater osmolarity. Of course, the optimal condition in which cells want to find themselves in is what we call an isotonic solution. Iso meaning same, tonic meaning solute, solution referring to the outside environment. So in isotonic solutions, the solute concentration of the inside of the cell equals that of the outside of the cell. And as such, water does move, but is going to move at a relatively consistent rate in and out of the cell. However, we do sometimes get into situations where we see things called hypotonic solutions. And in hypotonic solutions, we have the word hypo meaning less, tonic meaning solute, solution meaning the environment. And in this case, the cell has a greater osmolarity as opposed to the environment. And as such, water will move into the cell, potentially swelling that cell up and bursting it. Now, with animal cells, that's true. However, in plant cells, because of their plant cell walls, this actually is a good thing, because when this happens, the plant cell walls can limit the amount of water that can come in, and it will simply become turgid, looking very fresh. Now, the opposite phenomena happens in what we call hypertonic solutions. Hypertonic meaning greater solute concentrations in the solution, which is the environment. And in this case, water will move from the cell to the outside of the cell, as the outside has greater osmolarity. And in both plants and animal cells, this will result in shriveling of the cell, thereby killing it. And specifically in plants, this is referred to as plasmolysis. One critical example that AP biology loves to talk about is paramecium. Paramecium are single-cell protists that live in freshwater environments. And because they're cells and because the environment is fresh, they themselves are hypertonic to the environment, or we have a hypotonic solution in which the paramecia reside. Now, in this case, because water is going to continually move into the cell, paramecia have to do something in order to deal with the influx of water. So they've evolved a very special vacuole called the contractile vacuole that fills with water and utilizes energy in squeezing that water out consistently. Now, let's get back to membrane transport of materials, because the last one is about active transport. So not passive transport, but active transport. Now, in active transport, we're moving against the concentration gradient. And cells definitely might want to do this if they want to move things in and out of the cell against their concentration gradient. Imagine a cell living in an environment where there's toxins. These toxins start to slowly seep in through the membranes of the cells. Now, of course, there is a greater concentration of toxins on the outside of the cell and a lower concentration on the inside of the cell. But regardless, cells are going to want to move these toxins out because it's bad for you. And the same example could be applied in the opposite direction if it's something that cells want to move into the cell against this concentration gradient. Now, one thing to note about active transport is that all active transport requires energy because you're moving uphill against that concentration gradient. Now, there are two main types of active transport pumps that we learn about in AP biology, and one is called a sodium-potassium pump, and the other is called a proton pump. Let's start with the sodium-potassium pump. The sodium-potassium pump is a dual-action pump, as in it allows the movement of 3 sodiums out of the cell and 2 potassiums into the cell with a single revolution. And of course, it uses ATP in order to perform this action, ATP being a currency of energy within living organisms. Now, the sodium-potassium pump sets up what we call an electrochemical gradient. Now, electrochemical gradient can be a little bit confusing, so let's break it down. Electrochemical. Let's start with the chemical gradient. If you have sodium-potassium pump operating, you are going to build up a larger concentration of sodium on the outside and a smaller concentration of sodium on the inside. On the other hand, you're going to have a larger concentration of potassium on the inside and a smaller concentration of potassium on the outside. This is what we call a chemical gradient. Now, what about the electro? The electro refers to the fact that because you're pumping 3 cations out of the cell, but pumping only 2 cations into the cell, there is a deficit of 1 cation on the inside of the cell. And as a result, the inside of the cell membrane tends to be slightly negative, while the outside tends to be slightly positive. We call this slight negativity a resting membrane potential. Let's talk about the proton pumps. Proton pumps are also going to create a slight negativity on the inside because in this case, we are pumping out protons, or what we call hydrogen ions, to the outside of the cell via the use of ATP. The reason that we learn about proton pumps is that while the proton pump is what we call a primary active transport pump, it's associated with another protein called a sucrose proton cotransporter, which is part of what we call a secondary active transport. And the reason that is secondary is that with the proton pump, we're able to first establish a chemiosmotic gradient of protons on the outside of the cell. And as we said earlier, when you have a lot of protons on the outside, they want to come back in. And as they come back in, it releases some of that chemiosmotic potential into energy that can now move sucrose against its concentration gradient from the outside to the inside. So basically, it's coupling energy in many different ways. We start off with ATP, which is chemical energy. We transform that into a chemiosmotic potential, which is energetic. And then through the falling action of those those protons back into the cell, we're able to drive this protein into moving the sucrose against its own concentration gradient, resulting in what we call transport energy. And that about sums up transport of single materials in and out of the cell. Now, there is this whole other section on exocytosis and endocytosis, and I think those are important, obviously, because that's part of your AP curriculum. However, the idea of bulk transport is not too difficult to understand, and I'm going to link to a video which really shows many great examples of vesicle formation and vacuole formation in both exocytosis and receptor-mediated endocytosis. So watch those videos, and I think you'll have a better understanding of that. Otherwise, that pretty much sums up the contents of Chapter 7. And of course, the primary source for information is going to be your textbook, but I hope this video somewhat summarizes that chapter well for you and that this has been helpful. If you like this type of content, consider subscribing to our channel because we're going to be producing videos on a weekly basis, if not more, and like and share this video with your friends because we're all in this together. And once again, it's been great having you guys here today with me. My name is Mikey. Have an amazing day.